Hi, I'm Jen Fisher, and the objective of my study is to evaluate different dimension reduction methods for the application of transfer learning. So, rare diseases have limited data sets. Due to the nature of rare diseases having small number of patients, this can be a problem for statistical and machine learning methods where we try to identify drug repurposing candidates that can help with the sad statistic that 95% of rare diseases do not have a molecular target for therapy. And so what transfer learning does, it learns patterns from large statistically powered data sets and applies it to smaller data sets that aren't powered enough to do the analysis that we're interested in. And so for this project, I looked at several dimension reduction methods as this is a precursor to transfer learning to identify patterns in the large data sets. And I looked at principal component analysis, independent component analysis, and non-matrix factorization. Using these methods to identify latent variables that I apply to cancer data sets, including the Cancer Cell Line Encyclopedia, the Cancer Genome Atlas database for glioblastoma, and then compared the two databases with the Spearman correlation of the different latent variables to see how well the profiles correlated before and after transfer learning. I also did a pathway analysis on the latent variables calculated and see which KEGG and reactant pathways were enriched. So now to go into the results. First, looking at principal component analysis. So this first heat map here, we have the PCAs on the y-axis, and then on the x-axis we have the different samples from the Cancer Genome Atlas glioblastoma data set. And so these samples are colored by if they're recurrent, they're red, if they're primary tumors, they're blue, and if it's a normal sample, it's in green. And so the principal component analysis did a really good job of separating out tumor versus normal. The second heat map here is of the Cancer Cell Line Encyclopedia glioblastoma data sets. It's not surprising here that we see a lot of heterogeneity with glioblastoma, as that is a characteristic of the disease. However, it is interesting with this PCA plot heat map that we have cell lines that are clustering together. Next, we did an independent component analysis. So some people think that independent component analysis is not appropriate for biological data due to independent component analysis assuming that you have independent signals from your data set. However, when we look at independent component analysis at a K of 10 and a K of 50, the clustering algorithm based off the independent component scores identified clusters for normal and versus tumor, so this might be an indication that independent component analysis might have an application in biological context like transfer learning. Lastly, we looked into non-matrix factorization, and it was also able to, along with PCA and independent component analysis, identify differences in clustering between tumor versus normal. We also did a pathway enrichment analysis for the different latent variables to investigate what pathways were associated with the different latent variables. And some of the latent variables shared pathways, and some of them had unique pathways. So we were interested to see what the distribution of these unique and shared pathways were between the different latent variables to see if there was a trend of unique versus shared pathways between the different latent variables. Lastly, we did Spearman's correlation between tumor profiles and cell line profiles. So on the x-axis, we have the cell line profiles. If the cell line was of glioblastoma, it was black, and then if it wasn't, it was gray. It was another cancer type. And then on the y-axis, we have the tumor and normal profiles from glioblastoma. So overall, there's pretty good correlation between the tumor profiles and the glioblastoma profiles as they cluster together. While when we look at the transformed profiles, they're less correlated, and this leads to the clustering dispersing for glioblastoma cell lines. However, it's still interesting to note that tumor versus normal still separates out on the y-axis, and this trend continues with PCA and non-matrix factorization. So this leads to avenues in the future to improve these dimension reduction methods to improve the correlation between tumor profiles and cell line profiles that was seen with the transcripts per million. Overall, the study, we were able to determine that most of the methods with various numbers of latent variables were able to distinguish between tumor versus normal. It was also found with the dimension reduction method pathway analysis that it seemed like non-matrix factorization had more KEGG pathways and more unique KEGG pathways for latent variables calculated than the other methods. However, it's hard to compare non-matrix factorization latent variables from higher K groups, as those weren't calculated for this study. There's also the potential to use various dimension reduction methods for transfer learning applications. And some future directions from this project include drug repurposing applications with transfer learning, as the Cancer Cell Line Encyclopedia does have information about how those cell lines respond to drug therapeutics, and we can also apply transfer learning to other drug repurposing databases. Also, there's the application to evaluate dimension reduction methods outside of cancer and other biological disease contexts. We could also use ReCount3 database to create other latent variables, as ReCount3 is a larger database compared to ReCount2, which we used for this study. And so I just want to finally acknowledge my lab, who have been very supportive of this project and super insightful, my committee, and funding resources. Please let me know if you have any questions. I'm very excited to talk about this topic with others here at this conference.